Assalamualaikum and good morning. My name is An Wahib and today I'm going to be presenting on my proposal for my final year project research, which is entitled Microplastics in Bottled Water and its Health Risk Assessment in Malaysia. I also have my iPad down here, so if I were to look down, it's for my slides. So for the introduction, first, microplastics are found in freshwater system. Before this statement, microplastics are commonly found in marine waters, such as the ocean. However, previous research has shown that microplastics can also be found in freshwater system, such as rivers and lakes. This could be sourced from insufficient water treatment plants, which causes presence of microbeads from personal care products to be present in these freshwater systems. These microplastics, when they are present in the environment, could bring several effects. First, the primary effects would be causing blockage of bloodstream of aquatic animals that are in the environment. And the secondary effects, which are the release of additives such as plasticides or phthalates when these microplastics degrade, and these additives have EDC properties. And these microplastics could also be carriers of hazardous chemicals and heavy metals. Now let's move on to the problem tree analysis. As you can see, the core problem is microplastics found in bottled water. The few of the causes would be, first, the inefficiency of wastewater treatment plant and drinking water treatment plant, causing plastic waste to exist in the freshwater system, and these plastic waste could then degrade when they are in such environment. And the other cause would be the low quality of plastic packaging or bottles that are used in the commercialized bottled waters. And these low quality plastics could go through degradation and could cause presence of microplastics in the water that we drink. And this core problem could cause certain effects, and a few of them would be chronic exposure of primary and secondary effects over a long period of time, and the quality of bottled water could be questioned, therefore it could decrease in sale. And then we move on to the problem statements. I've listed 3 of them. So first, it would be heavy metal contamination of raw water sources. This could cause the increment of bottled water purchasing, and the increment of buying these plastic bottles, buying this bottled water could cause the increment of plastic waste thrown into the environment if they were not recycled properly. And this leads on to the second problem statement, which says plastic bottles could degrade under natural conditions through photooxidative and hydrolytic pathways. And when these plastic bottles degrade, they produce microplastics, and these microplastics can cause possible defects to one's body. This would be the SWOT analysis for my research. The strength of the research is that it consists of niched sample sources of the sample that will be included in the research will only come from Malaysian produced drinking bottled waters. And the research too includes calculation of hazard ratio for microplastics concentration. However, on the other hand, the weakness of these research is that secondary risks are not analyzed. And the methods used in analyzing the microplastics present are chosen, as the methods for analyzing microplastics have yet to be standardized. The opportunity of this research is that the output consists of data recordings for a specific country. And my research too could spread the awareness on the microplastics presence in drinking water alongside prior research. However, the threats present would be this research does not include nanoplastics potential effect, as it focuses more on the microplastics presence, and because of the weakness of the research, it causes this particular threat, which is the secondary effects of microplastics are still unclear. These are the objectives for my research. First, it is to ascertain the physical and chemical properties of microplastics in bottled water. And second, it's to appraise the risk level of microplastics ingested from bottled water to the populations in Malaysia. The research questions are as so. What are the type, color, morphology, and size of microplastics present in bottled water? What is the risk characterization of the microplastics level in bottled water? And what is the major pathway for microplastics to reach the population of Malaysia? These are the significance of the study. First, it could increase the awareness to the public that the waste that you litter goes back into your body. This study could also fill the research gap for further microplastics inspection in regards of secondary effects. It could also alert the public on potential risks from ingested microplastics. The data obtained could act as catalysts for the betterment of future water treatment plants. And this study too could give updates to the environmental public health sector on new microplastics pathways. The slide is now showing the literature review included in the proposal part of the research. Dijkstra et al. 2014 have proved that drinking water sources come from tap water, groundwater, and recycled water. And tap water and groundwater are a part of the freshwater system. Orton et al. 2017 have proved that microplastics have indeed been found in freshwater system and terrestrial environments. Therefore, putting up a notion that microplastics could also be present in drinking water sources. Relic et al. 2017 have proved that microplastics from surface could be transported or seep into groundwater. Therefore, suggesting that microplastics could also be found in these bottled water. This research by Mason et al. Entitled Synthetic Polymer Contamination in Bottled Water has proved that in 259 bottled water samples, the mean concentration of microplastics found is up to 10.4 microplastics per liter. Therefore, proving that they are significant concentration of microplastics found in bottled drinking water. Research by Wang et al. 2020 have mentioned that drinking water treatment plant are able to remove up to 82% to 88% of microplastics from raw water system. And the remaining microplastics that are still present in the effluent would be discharged into the nearby freshwater system and could accumulate over time. Research from Lee et al. 2018 has proved that polymers do degrade and the bioaccumulation of additives could happen. And as the analysis of microplastics in bottled water uses the principle of spectroscopy, these would be the most common spectroscopy apparatus used in prior research. So first, it will be FTIR spectroscopy. This apparatus could identify carbon-based polymer. Therefore, it could distinguish plastic from other particles. SEM spectroscopy could form image of microplastics without additional installment. However, software is needed to aid in identifying C-dominant plastics, and this software is called the EDS. Raman spectroscopy could identify microplastics at a small size, and it could ensure the initial condition of the particles in tests, as it uses the no contact policy, or no contact technology. Right, so moving on to the methodology framework. As shown, the preparation part of the methodology would consist of continuous background study, literature reviews, and methodologies comparison, and then the materials availability will be checked, and sample planning. Then the methodology will be chosen based on materials availability and sample size. It would consist of a collection and preliminary preparation of samples. Samples preparation would consist of blank preparation, mineral digestion, and sample dyeing with Nile red solution. Next, the samples will go through filtration and analysis with FTIR spectroscopy, and then the calculation of microplastics concentration and risk characterization will be done. Next, the data will be statistically analyzed that could prove the reliability of the objectives mentioned. These are the steps in the methodology chosen. So the first part would consist of chemical analysis. So the blank preparation would be ultra pure. Would be samples of ultra pure water, and the sample will be water samples from bottled water. And these samples will go through mineral digestion using EDTA solution, and the samples will be dyed using Nile red solution. Next, SDS solution will be added into the mixture for particle suspension and will be filtrated. The filtered paper will then be analyzed in FTIR for the inspection of microplastics particles. So the concentration of microplastics found in 250 milliliter samples would be extrapolated to 1 liter sample, as this information would be included in the calculation of risk quotient. These would be the expected outcomes of the research. First, significant amount of type 1 plastic would be found in each bottled water sample. The primary risk of microplastics concentration would be considered as low. New pathway of microplastics to human could be deduced. This slide shows the Gantt chart, and there will be 4 milestones throughout the research process. The research activities to be conducted would be similar to the methodology framework mentioned before, and the estimated period for completion would be approximately about 4 to 5 months, and hopefully, if I were to be able to start in May, it would end in September 2020. So this would be the reference list. That will be all. Thank you, and stay safe.